everyone. This is Courtney speaking. Our presentation today is on something called Salivaomics, and we're going to go over that with you. We're going to explain, not that you guys already don't know what saliva is, but we're going to explain the components behind it and why this is a good tool used for diagnostics. And we'll also go into what the omics technology is and also some other key components that goes into this technology and how it's used how it can be an alternative and an easier way to diagnose certain things within the mouth. Part of our group is me, Courtney Bryce, and then there's Mohammed.
Hello, how's it going?
And Nida's with us.
Hello.
Sana.
Hi, everybody.
And Valerie.
Hello, everybody.
All right. So what is saliva? The definition of saliva is it is a complex oral fluid that includes secretions from 3 pairs of major salivary glands, and those are parotid, submandibular, and sublingual. And from the minor salivary glands, you have labial, buccal, lingual, palatoglossal, and palatal mucosa. The gingival crevicular fluid, cellular and food debris, bacteria of the oral cavity and their metabolites, as well as upper airway transudate. The functions of saliva, which all of these we already know, but so the main functions of saliva are protection of the oral cavity from infections such as caries and promotes swallowing and degradation of ingested food. Buffering action by means of bicarbonate and phosphates in protein, lubrication for the mouth, remineralization of enamel by the rich proteins, staurin, calcium, and phosphates, and it also helps with the inhibition of demineralization associated with mucins. More behind the compositions of saliva. Saliva mainly consists of water, which accounts for 99% of its composition, and the other 1% is a complex of organic and inorganic molecules. Saliva's main organic components consist of proteins, enzymes, mucins, and immunoglobulins, of which secretory IgA is the most abundant and has long been recognized as the first line of oral host defense. And another composition is that saliva consists of salivary proteins with amylase, lipase, proteases, and mucins, which facilitate digestions, while lactoferrin, lysozyme, lactoperoxidase, and histatins exhibit antibacterial properties responsible for hydrolysis of cellular membranes and interference with the binding of microorganisms.
Once again, hi, everybody. I'm going to go over why is saliva used for diagnosis. So basically, collecting saliva is non-invasive. It's inexpensive, less technique sensitive, and easy to perform as compared to serum. So we all know with blood testing, it's going to be needles and puncture and pain, but with serum, with saliva, it's really easy to get it. And then saliva is found for detection of oral diseases like periodontal disease, caries, oral cancer, salivary gland disorders, and non-oral distal diseases, maybe like systemic diseases. So what basically is omic technology? It's primarily aimed at the universal detection of gene, mRNA, proteins, and metabolites in a specific biological sample. So just like in blood sample, there are many things involved, and we check for their levels if, for example, red blood cells or white blood cells or any platelets or anything, and we check for the detectable changes and see if there's any disease condition present or not. So similarly, this omic technology is used using saliva, and we can check for various diseases or disorders or maybe any risk factors. So what are the advantages of this omic technology? Disease states may be accompanied by detectable changes. It is easily collectible. Storage is really easy, shipping and voluminous sampling than serum or urine. So it has many advantages over blood testing or urine sampling. But there's also some limitations, and one of the limitation is with this testing, we cannot always diagnose some systemic conditions like diabetes because additional testing is required. So limited research on linking substance in saliva to some systemic diseases as other tests are needed to confirm diagnosis. So alone, the saliva testing cannot be used. So Salivaomics has 5 diagnostic alphabets, which are proteins, mRNA, miRNA, metabolic compound, and microbes. So basically, we are testing for all these components of saliva to check for any changes.
Hello, everyone. So I'll talk about the protein, one way of these 5 prognostics, and then the proteomics is the study of all proteins in a given sample. Saliva proteomic analysis includes the characterization of protein mixture in order to understand the complex biological systems and determine relationships among proteins, their functions, and the protein-protein interactions. Also, it can provide details on entire body's health because saliva is an excluded of blood and contains juices from gingival clavicular fluid along with major and minor salivary glands. And the second one is mRNA. The characterization of mRNA profiles in body fluids provides insights into gene transcription in normal and disease states. It have been pursued for a number of diseases, including OSCC, primary syndrome, pancreatic cancer, and ovarian cancer, and they have proved to be sensitive, specific, and accurate. There are 3,000 human mRNAs in a cell-free saliva supernatant of healthy subjects. Particular interest in is that there is a normal saliva transcriptomic core signature of 185 mRNAs that is present in all normal subjects, providing the rationale to use the salivary transcriptomy for disease detection. Then it is miRNA, microRNAs are they're short parts of RNA transcripts are more specific and potent compared to mRNAs. It exhibit high sensitive, which effectively detect and differentiate poorly differentiated carcinomas.
Hello again. The fourth diagnostic alphabet is metabolic compound. Metabolics is the global assessment and validation of endogenous small molecule metabolites within a biologic system. Also, metabolites are important indicators of physiological or pathological states and can provide information for the identification of early and differential markers for disease. It also helps to understand its occurrence and progression. Biomarkers plays an important role in monitoring the state of biological organisms and is widely used diagnostic tool for disease. Lastly, we have microbes. It provides a clearer picture on diagnosis and find out the severity of the oral disease. It enabled the detection of various oral infection diseases as well as upper respiratory infections caused by influenza virus, human Boca virus type 1, and infections by cytomegalovirus, uh, eh, Epstein-Barr virus, human herpes virus, and Zika virus. Another test, aside from the 5 diagnostic alphabets, is CARE test. So its function is to test a host susceptibility to caries. Test, it's a test to see if certain oligosaccharides are present in a patient's saliva. If certain oligosaccharides, which are preferred or not preferred by oral carcinogenic bacteria, are present or absent in host saliva, it will likely lead to increased risk for caries. So for the advantage, advanced knowledge of who is more susceptible to caries, and this can help prevent caries. For the limitation, these tests sometimes cannot be applied to everyone because it does not take into account other factors that may cause caries.
So the two ways that the saliva is collected by is A would be stimulated saliva is collected by masticatory action or by gustatory stimulation. And the other way would be unstimulated saliva, which is collected without exogenous gustatory, masticatory, or mechanical stimulation. Okay, and so the best two ways to collect saliva would be draining. Draining method is in which saliva is allowed to drip off the lower lip, or spitting. Spitting method includes in which the subject expectorates saliva into a test tube. Okay, and saliva storage. Saliva is very sensitive as it fluctuates with time of collection, the pH level of like the surrounding environment, temperature, type of saliva, types of saliva collection method, and storage method. Storage is usually kept at 80 degrees Celsius, provides less biochemical and microbial changes in saliva, hence gives better results when it's compared to 20 degrees Celsius. To avoid altered result, there should be a time gap between sample collection and analysis. So in conclusion, saliva is a useful diagnostic tool and can be used for early detection of systemic and oral disease. And because of the ease of collection of saliva and due to it being very inexpensive and non-invasive, collection method make it more compliant compared to blood sampling, as it's especially for the kids or adolescents, which may be afraid of needles or blood or so on. And but one thing is more and more tests are being developed to use saliva as the testing means to diagnose oral disease or to test the susceptibility of oral disease. Example, the CARE test where we went over, or NIDA went over, how it basically detects how prone someone is to cavities. But more research needs to be done on to use saliva as a diagnostic tool for systemic disease, as it compares a couple of studies where some proteins are there for diabetic patients or compared to a healthy patient, or some proteins are available in kids with autism, but that still isn't a test that you can rely on. So there's still more testing that has to be done. So saliva is a good tool, but it doesn't give a definitive diagnosis for a systemic disease. And that is the presentation on Salivaomics.
If anyone has any questions, please feel free to put your questions into the chat in the Zoom, and we'd be happy to write back to answer any of your questions that you have. Thank you for listening, and take care. Bye.
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 Have a good one.